Good evening, dear students. Today I'm going to tell you the most important topic from biology class 12th, that is from the chapter Molecular Basis of Inheritance. Right? In this, I am going to explain the answer of the question like experiment conducted by Alfred Hershey and Martha Chase for identification of genetic material. And the other part of this question is, why is it considered path breaking in the field of molecular biology? The experiment was conducted by Alfred Hershey and Martha Chase, and the objective of the experiment was to show that DNA and not protein is the genetic material in the bacteriophages. More precisely, we can say that this experiment is done to show that DNA is the genetic material, not the protein. So what they did, they grew two different sets of bacteriophages. In one, the DNA was made radioactive with 32 phosphorus, and in the other, the protein coat was made radioactive with 35S. Protein coat, okay, 35S. These phases were allowed to infect E. coli bacterial cell. Soon after infection, the cultures were gently agitated in a blender to separate the adhering protein coats of the virus from the bacterial cell. The phase means a kind of virus, bacteriophage that attack on the virus. So this phase bacteria is invaded by bacteriophage. So bacteriophage is a kind of virus that attacks on bacteria. The phage injects only its DNA into the host of bacterial cell. The cultures were then centrifuged to separate the viral coat and the bacterial cells. The culture out here is, in biology or in chemistry, there is a word called supernatant. It is actually a kind of liquid on which a residue is present that has been separated out from the culture. That is called supernatant liquid. It was found that when phase containing radioactive DNA was used to infect the bacteria, that was 32P, the radioactivity was found in the bacterial cell, right, which were found in the sediment, indicating that DNA is the genetic material of the phase and not the protein. This experiment showed that DNA is a genetic material that passed into the bacterial cells. They also showed that protein is not the genetic material, as protein as S35, which affects the protein, not even enter the bacterial cell.